Hey, everybody, and welcome to Unit 6, Lesson 1, Exploration 2. And so today is all going to be about DNA structure and how it's going to work together, how all living organisms are going to share the same structure of DNA, and that's how we're going to be able to compare organisms and species together. But we're going to look at some of the scientists who helped discover the structure of DNA and their journeys, but then also what is the exact structure and how are all these species going to use the same molecule to pass on traits. So, we talked about Hershey and Chase in the last unit, and also Griffith and some of the other people that helped discover kind of to lead us up to that there is something passing on traits from parent to offspring. And so now we're going to look into DNA. So the first thing you need to understand is DNA is going to be made up of building blocks. And these building blocks are called nucleotides. So it is a monomer that forms DNA. It has a phosphate group. It's going to have a deoxyribose sugar, and it's going to have one of four nitrogen bases. And we'll talk about those four nitrogen bases today. So one molecule of human DNA is going to contain billions of nucleotides, and you'll see how this works. But basically, the phosphate and the deoxyribose are going to be the same in every single nucleotide. And we're going to see that later when we talk about the backbone of DNA. But this nitrogen base is what's going to be the language of DNA. So there's one of four nitrogen bases, and down here we'll see that they are cytosine, thymine, adenine, and guanine. And you'll hear me talk about A, T, C, and G, right? A, T, C, and G. Those are the nitrogen bases: adenine, thymine, guanine, and cytosine. So it can be one of those four. And based on the structure and based on the order of those nitrogen bases, we are going to be different. And that's the difference between you and me and any other living organism, from a plant to a bacteria to anything that's going to have DNA. What makes them different from you and me is the order of these nitrogen bases. Okay, so let's take a look at these. We're going to break them into two categories. One of them is called pyrimidines, and the other one are going to be called purines. And the way they get broken up is by their structure. So what you can see here is that thymine and cytosine are going to be our pyrimidines, and they are going to have a single ring, right? Because over here you can see that these are going to be made up of two rings. And so what we're going to find out is that we are going to have a pyrimidine and a purine match up across from one another. And that is going to give us a total of three rings, right? And what we're going to find out is that A and T are always going to match. And we'll talk more about this later. But when thymine and adenine get across from one another, they're going to be three rings. And also cytosine and guanine. So cytosine is also a pyrimidine, and guanine is a purine because it has two rings. And they are going to get across from one another and give us a three ring makeup too. And that's very important. Okay, so what we need to know here is these are our nitrogen bases, right? Adenine, thymine, guanine, and cytosine, and that our pyrimidines have a single ring and our purines have a double ring. Okay, so this chart is going to be very important for your test later on. As we scroll down, it says which base is most similar to the structure of thymine. So if we look back up here, we see that thymine is a pyrimidine. Cytosine is also going to be, oh my goodness, excuse me, sorry about that, is going to be a pyrimidine as well. So that's going to be the one that's similar. So determining DNA structure. So how did we get DNA structure? There's going to be a guy named Erwin Chargaff. And what Erwin Chargaff is going to do is he's going to run tests on DNA from a bunch of different species. And here are the different species that he looked at. And what he did is he broke down DNA into the nitrogen bases, into nucleotides: adenine, thymine, guanine, and cytosine. And what he was able to determine is when you compared the DNA, if you looked at the adenine and guanine, they were 1.5. They weren't consistent. And when you took thymine and cytosine together, they were not consistent either. However, when you compared the number of bases from adenine and thymine, A and T, they were almost right at 1. And we're talking about, billions of nucleotides that he's comparing, not just a few. We're talking about a high sample size. And also, when you compared G and C, guanine and cytosine, they were very similar. And when you compared purines to the number of pyrimidines, so basically we did remember, we go back up here and we look at it. Thymines and cytosines, when we compared those to adenines and guanines, they were also almost 1 to 1. And so what Chargaff was able to conclude is that there are relationships in these nitrogen bases, and that some reason adenine and thymine have a real relationship, and guanine and cytosine have a real relationship, and then we're going to see a relationship to the number of purines and pyrimidines. And what you're going to find out is every time we have a purine, we're going to have a pyrimidine across from it. And every time we have a guanine, we're going to have a cytosine across from it. And adenine and thymine is the same. So they're going to have a very close relationship. The next person we want to talk about is a woman, a British scientist named Rosalind Franklin. And Rosalind Franklin is an individual who worked with X-ray photographs and X-ray, what we call crystallography. And what she's going to do is she's going to use X-rays and run it through DNA. Here's a picture of her on her microscope. But this is one of her pictures, photo 51. And this photo, if you are a scientist and you're studying X-ray crystallography, when you see this pattern, you know that this is a helix or a helical formation. And this was important because we know that the primary structure for DNA is a double helix. And she's going to have this picture, but two other gentlemen by the name of Watson and Crick are going to get this picture. And it's going to make a lot of sense when they compare it with their information that they have as well. So as we scroll down, here is Watson and Crick. So it says about the same time Franklin was working with crystallography, right? Watson and Crick were working on structures. They were using models. And so their model that they were building, they were using nucleotides, but what they did is they had the phosphate and sugar backbone on the inside and the nitrogen bases on the outside, which they were at a wall. They were not making any progress. Well, then they saw Rosalind Franklin's photograph here, and that kind of showed them that the nitrogen bases were going to be on the inside. So what they did is they put the phosphate and the sugar on the outside, and that's going to create what we call a backbone. Okay, the sugar phosphate, they put it on the outside, and they put the nitrogenous bases or nitrogen bases on the inside. And they knew from Chargaff's work that A and T were going to go together, and C and G were going to go together. And so therefore, they were able to kind of discover, right? It says right here in 1953, they published their work. And this is where they were able to kind of take credit for the discovery of DNA. Now, we also, when we talk about them, and this is them right here, they were able to get a hold of photo 51 from Rosalind Franklin through a mutual friend, and kind of she did not know. So it was kind of sketchy. But either way, these two men were contributed with the Nobel Prize for the discovery of DNA. However, and by this time, Rosalind Franklin was well known, and it was known that they used her information. But in order to get a Nobel Prize, you have to be alive. And Rosalind Franklin had passed away because of all of her work with X-rays. As you know, X-rays, when you are standing around them, not only do they go through what you want, but they also are going to go through what you don't want. And so her body, by the time she had worked with X-ray crystallography for a long time, her body was just riddled with cancer, and she died at a very young age and was not able to get the Nobel Prize for the discovery of the structure of DNA, even though she was a leading contributor. So here's the current model of DNA. So here's a nucleotide right here, right? Phosphate, sugar, and then the nitrogen base. Here's another one, and here's another one, and here's another one, and so on. And here's one over here, and over here, and over here, and over here. And so what we get is we get A and T across one another, and G and C across one another. And those were the things we talked about earlier. So we have nucleotides linking up, okay? We're going to get a bond on the outside, and we're also going to get a bond across the middle. These are the hydrogen bonds that are across the middle, and when you unzip them, which we'll talk about later, you're going to break these hydrogen bonds, okay? So this is the primary structure of DNA. We're going to have nucleotides are going to link together, right? There's 4 nitrogen bases, A, T, C, and G. And A and T, sorry for the bell, A and T always go across one another, and C and G always go across from one another. So this is going to make it more consistent when we go to talk about replication. Okay. So once again, that is going to be the end of this exploration, and I can't wait to see you guys in class.